Hi, my name is Max. I'm happy to introduce the CortXplorer research group at the Leibniz-Institut for Neurobiology. We are a multidisciplinary team of neurobiologists, psychologists, mathematicians, and computer scientists. It is our mission to understand how our brains work during decision making and learning. We want to specifically find out how neurons in their networks talk to each other while memories are established. We combine different techniques ranging from electrophysiology, learning tasks, up to genetics, molecular tools, up to computational modeling.
We're going to get a little more into detail about our specific projects now. We all currently research auditory learning and memory. To help us, we have Yo-Yo here to talk about what we do and maybe ask some questions.
Hi, thanks for having me. I'm Yo-Yo, and I'm here in behalf of the Long Night of Science audience to ask some questions about everyone's research.
Hi, I'm Katrina. I'm from the US, and I'm working as a PhD student in the CortXplorer lab at the Leibniz-Institut for Neurobiology in Magdeburg.
Hi, I'm Marina. I'm a doctoral researcher at the Leibniz-Institut for Neurobiology in Magdeburg, and I work also in the CortXplorer lab.
Hi, my name is Jing. I come from China and I work now as a PhD student in the Leibniz-Institut in the Department of System Physiology of Learning and Research Group of Comparative Neuroscience.
I look at how sounds are understood and represented by the basic units in the brain, neurons. Billions of neurons form hundreds of thousands of organized communication networks. In the auditory cortex, they organize in a clear and efficient way. We can change how single cells work and watch how that affects the entire network. Neurons perform large calculations of inputs in order to decide if they should send a signal to the next part of the network. I can modify a small part of this calculation and cause cells to always send the signal on. I do this by making a small part of the cell sensitive to light, and then I shine light on the brain to activate it. I'm interested in learning how network activity works in this way. When we understand how neurons communicate and what all the parts of the network and cells do, we can understand everyone's brains better and possibly help develop solutions to hearing loss.
So, as I understood, is that your research, Katrina, focuses on when neurons activate and don't activate in the auditory part of the brain. Can you elaborate on that? Like, how do I have to imagine that process? Why do they activate or not activate?
Neurons exist in large populations. There's hundreds of thousands of neurons sitting together and communicating with each other. And they have these spiky branches that receive signals, and they also have spiky branches that send signals out. So a single neuron takes in thousands of inputs, and it basically uses these inputs and where they're from and how strong they are to calculate if it should send the signal farther down the line. So you have a lot of really intense statistics going into whether a neuron will fire or not. That is to say, send the signal on to the next part. And that's the part that we're trying to gain control over so that we can say if the neuron receives a signal, it also sends a signal. So it should increase the activity that we see of the neurons in the population.
Thanks for your answer. Another thing that I am curious about is that you said you're making some neurons are a part of the brain light sensitive. How do you do that?
That's a really interesting question. It's actually a tool called optogenetics that a lot of people use in different ways. Basically, we inject the brain with a protein that targets the cells, and then over a few weeks, it changes the DNA of the cells that it gets inside. These proteins were originally discovered as in plants and moss that respond to light, and so they're able to actually genetically change the neurons in your brain to also respond to light.
Then how do you make sure that what you inject into the brain targets the cells that you actually want to target and not spread into other parts of the network that you don't even want to like react to light?
That's a really good question. There's actually two things that happen when we're injecting the protein that help us determine where and in which cells it is. The first is that the protein is actually injected into the auditory cortex, the hearing part of the brain, and it can only go so far. So it can only spread through so much of the brain. It, in our case, really doesn't spread past the auditory cortex. The other thing is that we have a genetically modified mouse, because I'm using mice. This mouse is bred just with one extra protein that doesn't do anything, but that protein is attached onto specific cells, and it's like marking those cells with an X, so that when the protein is injected into the brain, it finds the marked cells, and it knows that those are the cells that it needs to go inside.
Imagine yourself staying at the traffic lights. You know very well that you can cross when it's green light and staying when it's red light. What will happen if one day, all of a sudden, this rule changes? You need to relearn crossing with the red and staying with the green. It might take some time, but at some point you will be able to adapt to this new situation. This is called cognitive flexibility. In my experiment, I used gerbils and I trained them to learn the meaning of specific sounds, like we know the traffic lights. While they learn and relearn, I'm looking at their brain activity and especially at the area which is processing sounds to see what exactly is happening.
Marina, you mentioned cognitive flexibility. How is that important to us humans or our everyday lives?
We live in an environment full of stimuli. Everything around us is a stimulus. And we need to learn and relearn continuously. And this is how exactly the animals do. And the cognitive flexibility is everywhere in everyday life. So I think this is an important aspect for scientists to look at.
So for your project, for your research, you used gerbils. Why is that, and how does that relate to humans?
Yeah, thank you for your question. This is very interesting and important to say. So every animal has a specific, or humans as well, have a specific frequency range that we hear. Gerbils happen to have the same or similar frequency range with humans. And this is very important because whatever we find out about the auditory cortex of the gerbils, maybe we can relate it closely with the human auditory cortex. Additionally, gerbils are very good auditory learners. They can easily adapt to specific experimental designs and learn the auditory tasks that we provide.
I do research on auditory adaptation in the brain of gerbils. We are all familiar with the adaptation phenomenon. When you put on a cloth, at the beginning you feel there's something new on your skin, but after a while you will just ignore it and don't recognize it anymore. I am curious about how in the auditory system the adaptation is functioning. I record the brain signals in gerbil while presenting auditory stimuli that is causing the adaptation phenomenon. Besides the experiment that I am conducting in animal, my colleagues in the research group of comparative neuroscience are also conducting relevant research on human and in cortex modeling. And together, we hope this would help us to understand more about this function.
Interesting. Jing, you talked about the adaptation function of our brain. Can you explain why we even have that, or why we need that in our everyday lives?
Thank you. That's a good question. Imagine our brain has a certain budget of attention and energy to pay attention to the environment. When there is always the continuous and safe stimulation, then we don't need that much energy on it anymore. Instead, we can more efficiently to use our energy to pay attention to the change, to the danger, to the risk in the environment.
So you're saying that you're recording the brain signals while you give an auditory stimulus. How do you do that?
Because I want to trigger this adaptation phenomenon, and it is triggered by a repetitively stimuli. Because the stimuli is always the same and coming at a certain time point, and then the brain starts to decrease the response and starts to adapt. So what I give is that I give a certain tone with a certain frequency and the same amplitude repetitively with a certain time interval in between.
When you talk about signals, what are those?
The neuron, they are talking with each other with electrical current flow, and we could measure this current flow changing.
But I was wondering, because the way you explain it, it's like basic reactions towards like stimuli everywhere, every second in our lives, kind of. And for what Marina gave as an example, it has more of a complex learning quality, because her example is that we specifically learn that the traffic light is red, and that has a means for our behavior, which we think about, which is, I think, a different dimension of that learning.
Yeah.
So it's like mine is always the same stimuli come. How does the brain reacts? But for Marina's, it's like when the new stimuli come, and when plus the new stimuli, there's a new rule behind it.
Right.
I think maybe this is also related to the fact that Jing is doing an experiment, which is the animals are anesthetized, and you really look at the clear processing of the signal, while mine is the animals are behaving. So I think this is also like how also is related with the whole environment around, you know, and also like the ability of the animals to learn and adapt to the paradigms that we provide. I think this is, but I think it goes really deeper if we say all this.
True.
That's perfect, because I just got that conversation and it's going in. So, so there you go.
You can make it.
I think that was great.